than our high dose, which was incredibly effective in the mouse, 3.3 times 10 to the 14th vector genomes per kilo. And then we picked an intermediate dose that was a half a log higher than our low dose. So these are the three cohorts. The primary outcome measure for this trial is safety, unequivocally, but we will be looking at secondary outcomes. The CHOP in 10 that I mentioned in the beginning is considered the standard for the field in terms of the best clinical measures of natural history of the disease, and it includes these measures: head control, writing reflex, truncal movements, rolling, ventral suspension, and standing. And it is a very powerful clinical tool. We'll also, of course, use time to invasive ventilation. This would be an end of the study if there was. This would mark the end of the study if there was a requirement for invasive ventilation or obviously death. For follow-up, we'll be using we've done standard treatments before with gene therapy. We generally follow patients at 1 to 4, 8, and 12 weeks, and then every 3 months for 2 years. So basically, this is our clinical trial, and be glad to address any questions and call on our research team here that I've introduced. And I think we have a very good team. We have very good preclinical data, and I think we have a very good tox plan to get us to clinical trial. So I'll stop there. Thank you.
Thank you very much. Let's go to the reviewers, and again, I want to remind the reviewers, please highlight both the satisfactory answers to your queries as well as the ones that are not. Dr. Chatterjee?
Thank you for that a really nice presentation, and this is very exciting, innovative work done by your team, and it's nice to see it here. The first question I asked was about the gene that was going to be delivered in the vector to the patients. It wasn't clear to me whether it was going to be the SMN2 gene to try to make it similar to the SMA234 or the SMN1. Your response was that it was going to be the SMN1, the wild type gene. Am I correct in understanding that?
Let me just clarify. So SMN1 and SMN2 are genes, and the piece in the vector is the full-length cDNA. It is actually derived from SMN2, but it has exon 7, so it's identical in function to a full-length SMN transcript, which is what comes from SMN1. So there is no difference in function of the cDNA or the messenger RNA when it's full-length between SMN1 and SMN2. So yes.
The cDNA contains that exon 7 region?
Yes, it does.
Okay. All right, thank you. Okay, so that clarifies that. The next question I asked was, since the AV9 construct was created in, the packaging construct was created in Dr. Kaspar's lab, whether it was identical to AV9Q14. And the reason I asked that question is because for a while there were a number of different sequences for AV9 in GeneBank, and I think the Wilson lab is in the process of correcting some of that. But AV9Q14 is the one that was sent out to most labs as AV9. So
that's the one that we did use, the one that we've gone to talks with, and the one that'll go to clinical trial.
Yeah. Okay. So I just wanted to know whether it was AV9. Okay. So that your answer is perfectly satisfactory. Okay, so my next question was in Appendix M. There is a question where we would like to see the number of genome copies per cell in the transduced animals and the transduced tissues. The only data that I was able to find in the submitted response was on the copy number of RNA transcripts per microgram of RNA. So you have since provided the genome copies from your, I believe, your primate data. I do still have a question about this. Was this vector delivered intravenously or intrathecally?
Intravenously.
Okay, so the primate data where you provided the genome copies per cell was after.
Okay, so there's two things here. So the vector genome copy number per cell was done on an intrathecal injection into pig. And actually I also have the spinal cord data on that, and it's one copy per basically it's up to one copy at most, can be 0.4 copies, so it varies between those two areas. The one thing that obviously that is not, when you look at that, you can have either 5 cells that are transduced with 2 copies or 3 copies, and 5 cells that are not transduced, so it's really a mixture. So relative to micrograms of RNA or relative to cell number, I'm not sure that it actually implies anything different between the two. So the monkey data is, which Brian can address, is done by intravenous. The copy number data is done by digital droplet PCR and is from intrathecal stuff.
And Dr. Chatterjee, it's a great question, and we are currently in the process of performing that for our non-human primate studies. I think it's very promising on the aspect that Dr. Burgess has this the PCR and the sensitivity up and running in his laboratory, and we're collaborating with our non-human primate samples to answer exactly the question and the good question that you raised.
Yeah, obviously I wanted to also find out what the genome copy number would be after the intravenous delivery, after it crosses the blood-brain barrier, or what there is at the blood-brain barrier at that age. But yeah, I think that data would be really useful to have.
Yes, and just to go on further, we are performing full biodistribution studies on the non-human primates as well, to look out over time as well. So I think that's going to be important information for exactly the reasons that you're bringing this
and
up.
I think the comparison with the intrathecal injections would also be very useful in the genome copy number distribution.
Absolutely. We do have indeed plans to be collecting that information also in non-human primates in the relatively near future for an intrathecal delivery in non-human primates as well.
Okay, thank you. So that answers that question. The next question I had was, I think it was a typo. One of the figures in Appendix M stated the 6.7 times 10 to the 13th vector genomes per kilo as the moderate dose when it really is the low dose, and I think that's being corrected. Okay. So, my next question was, I was wondering whether you were doing primate studies with a high dose, the 3.3 times 10 to the 14th vector genomes per kilo, primarily in terms of potential immune response related issues. But I gather from what you just said that those studies are underway.
So actually, we had the 6.7 times 10 to the 13th vector genomes per kilogram dosing for the safety studies in non-human primates, as well as low, medium, and high dosing in mice that we took to the FDA in our pre-IND conference calls. And a specific question of ours was, should we perform additional non-human primate studies going to a higher dose Now, I do want to make note that our non-human primates at that medium dose, or the starting dose, 6.7 times 10 to the 13th, showed no adverse events from any of the hematology or chemistry or blinded histopathology reading of the slides. The FDA in the pre-IND response was that no additional non-human primates were required, given and we took their suggestions to that and initiated the formal FDA-sanctioned toxicology and biodistribution studies in mouse only, and we've completed the 24-week time point at a contract research organization.
Yeah. Given that these are infants and they're unlikely to have pre-exposure to AAV, I think that's reasonable.
And just to make another point of that, and correct me if I'm wrong, team, we do still have plans to check the children for any presence of neutralizing antibody as well.
That would be done pre-treatment.
You could potentially have maternal antibodies
Yes,
still
exactly.
persisting. My next question had to do with AAV surviving primarily as an episome, and you're transducing infants who are going to be growing. And I understand from the written material you provided that SMN expression in all the tissues is really, especially in muscle, is also potentially important for correction. So I was wondering whether so as the cells divide, there is a likelihood that the episomes may be lost over time. And I was wondering whether you had, first of all, any data on, you probably don't, but maybe while you're getting this data, you might think about getting that genome copies per cell shortly after delivering the vector in your animal model, and maybe at a year, to see how much reduction of genome copies per cell you're observing in...
We have that in other clinical trials, and
Okay.
there's no reason, neither muscle nor motor neurons divide. So we really have a stable population of cells. So the loss from division is not an issue. I'll let Arthur or Brian address how many genome copies we had. When we looked at in our alpha-sarcoglycan trial, we looked at treatment and then at 6 months, and we had no significant loss of genome copies.
So at what time points were those?
That was at 6 months. Actually, we didn't have that was at 6 months, so I can't say whether we had any loss. So you're right. At 6 months we had clear copies and clear gene expression that persisted for 6 months. That was in muscle.
Okay.
And if you want to address how many copies we had.
I think, Dr. Chatterjee, again, it's a good question, especially as we start thinking about a program, years from now and developing programs. We actually have plans in place, collaborating with Dr. Burgess, to actually look at the vector genome within motor neurons, the primary target cell within spinal muscular atrophy patients over time, and I think we're going to take that into great consideration of extending those studies to look at even 1 year after how many copies are there. And the approach that we've worked out in studies is to actually utilize laser capture microdissection. So we'll be performing specific studies directly on the cell types of interest.
That would be incredibly useful information.
I'll just add to that, we have the laser microdissection up and running. We've done it for things, we've determined the amount of SMN that they produce, so technically that's not a problem for us to do, at least in the mice.
Great. Okay.
The only other thing that I would say is, at least, and it's a little controversial in the field, but we can get actually complete correction of the mice with expressions in only neurons, using Cree drivers. I'm not going to go into all the technical details. So the requirement for high levels of SMN above what SMN2 in external tissues is very debatable.
Okay, okay. Well, that's good to know. Yeah. Okay. Okay, my last two questions had to do with shedding. Your responses to Appendix M stated that you did not find AAV being shed, but these are very high levels of AAV that are going to be administered. In your response, you said that that referred to a different intramuscular delivery of a different vector, and that you are looking for AAV being shed in these intravenously delivered studies.
Kevin, you want to address that, the shedding issue? Because you looked into this.
Sure, yes. We're well aware that AAV is likely to be seen in the excreta, so that would be potentially plasmid, semen, which wouldn't necessarily apply in this case, stool and urine. Some of the most recent studies, which are out of the Factor IX group from St. Jude, both in primates and in humans, within the first 2 to 3 weeks post-intravenous injection of a self-comp type 8 in an older patient population, they're able to detect vector genomes in plasma and stool as the two highest levels. But once you get out to 2 to 3 weeks, these things seem to be settling back to below detectable levels.
So, yeah, so the gene itself is not toxic, so it's
Correct.
probably not dangerous, but we would like to try to prevent inadvertent transduction
Sure.
of family members.
Yeah,
so.
and the paradigm presents unique circumstances, right? Because these patients will require high levels of care, and caretakers are going to be exposed to these things. So, we think PPE and those type of precautions are going to be important.
And then also the disposal of the diapers or
Correct,
whatever probably have to be autoclaved or
correct.
whatever. Okay. I did have one question that I did not have in my, in the questions that I asked originally, and that has to do with your doses. The lowest dose that you're using, did prolong the mouse's lifespan, but not as much as your higher doses. So is it, I had a question about, is it really ethical to use that lowest dose? Why not just do
Well,
more
I think we've
patients?
wrestled with this question in every clinical gene therapy trial that we've done. When we go to the FDA, they really have harped on the issue of safety, and they want us to do a progressive clinical trial to assure that we have a safe product. From my point of view as a clinician scientist, I would love to follow your advice. I mean, if we could convince the FDA that we could go to 3.3 e to the 14th, I think I would be much happier. And we could test them out again on that. I mean, that's a possibility. But in general, they haven't been so receptive.
Yeah, because your results at the 3.3 times 10 to the 14 dose is
I know, they're
very
incredible.
impressive. They survive up to a year. That's
I agree.
Versus 35 days for your lowest dose.
I agree.
So that's.
I mean, I think the FDA, we had pushback from the FDA because these were infants, and they wanted us to be absolutely sure, and we wrestled with them on this issue, and they said, just don't forget that safety is the main issue, and we have to be confident of safety first before anything else. So
Yeah.
I agree with you.
Yeah, and the other consideration is that you will not be able to treat these infants with a second injection of AAV9 after that.
I mean, if that was a recommendation from RAC, we could probably deal with it better. I mean, but that's the problem. But your point is well taken, unequivocally.
Well, thank you. Those were all my the
Thank you.
questions that I had.